Minor red blood cell counts. Whole blood can be diluted with isotonic saline. This prevents red blood cells from lysing, but also facilitates their counting. We use a hemocytometer. A hemocytometer consists of two identical ruled chambers with 9 1 by 1 millimeter squares. When a cover glass of uniform thickness is positioned over the ruled area of the hemocytometer, a chamber of 1 millimeter cube is created. The center square is further divided into 25 smaller squares. These squares are used to count red blood cells. Red blood cells are counted on the left top corner, on the right top corner, on the bottom left corner, on the right bottom corner, and the center square. Each square is further divided into 16 smaller squares. These squares facilitate counting. All the cells of each individual square are counted. To facilitate counting, we will use the following guidelines. We will begin counting on the left top corner. That is usually distinguished by the 3 lines that are surrounding this square. We will continue counting all the cells moving towards the right top corner. Once all of those cells are counting, we will continue counting the second row. We will move now to the left corner until all the cells are counting. We will continue this zigzag pattern until all of the cells are counted. To avoid counting cells twice, we will use the following guidelines. If the cell is on the left or top line, we will count it. If the cells are on the right or bottom line, we will not count those. For example, we will count the cells in the first square. This cell is on the top line, so we will go ahead and count it. We will continue counting the cells in this square. This cell is on the bottom line, so we will not count it. We will move on to the next square. We will continue counting the cells. This cell is on the left line, so we will not count it. This cell is on the bottom line. We will not count those. Those cells will be counted later. We will move on to the next square. Now this cell is on the right line, so we will count it. We will move on to the bottom square. This cell we would count. This cell is on the right line, so we can go ahead and count it. We will move on to the next square. Now this cell is on the left line, so we will not count it. This cell now is on the top line, where it was on the bottom before, so we will count it. We will continue this pattern until all of the cells are counted. Because we diluted our sample, calculate the total red blood cell count by multiplying the total number of cells counted by the dilution. Then divide them by the number of squares times the volume. The volume of red blood cell squares is 0.004 millimeters cubed. Results are reported as number of cells millimeters cubed.